on to move to the next panel. This one's for the topic retinopathy and medical retina. I'd like to invite the judges for the same. We have Dr. Manoj Khatri. Welcome, Dr. Manoj. We have Dr. Chaitra Jaydev and Dr. Paritosh Kamdar.
Dr. Manoj Khatri and Dr. Paritosh Kamdar are here. They are the judges for this session.
Good morning.
Chaitra madam is not coming, so they will be judging. Only Dr. Manoj Khatri and Dr. Paritosh Kamdar are there.
All right. So moving to the presenters, our first presenter today is Dr. Zia Sultan Pradhan. I see you're there already. Dr. Zia, please switch your camera on. You can start sharing your screen. You have 6 minutes for the presentation. Yes, we can see your screen, ma'am.
May I start?
Yes, go full screen, madam.
Please, go ahead.
Full screen, please. Yeah.
Good morning. I'm going to describe a study that we have performed to improve our understanding of glaucoma in patients with retinitis pigmentosa. Now, there is a well-established association between retinitis pigmentosa and glaucoma. Western literature has shown between 2 to 12% of RP patients have primary open-angle glaucoma. However, the literature on angle closure disease in RP is limited to case reports and case series. So the first part of our study was to determine the prevalence of primary angle closure disease in patients with RP. This was a retrospective review of all RP patients over the age of 10 years attending our ocular genetics clinic. Patients in this clinic undergo a thorough history, a pedigree charting, and a detailed clinical examination. All patients found to have a shallow anterior chamber, high intraocular pressure, or cupping undergo examination by a glaucoma specialist, where a gonioscopy and detailed optic disc exam are performed. Visual fields and ERG are done where required, and every patient undergoes a macular SDOCT. In our clinic, we saw 618 RP patients during the study period. Of these, 1.3% had primary open-angle glaucoma and 2.3% had primary angle closure glaucoma. Hence, the first thing to note is that angle closure disease is more prevalent in the RP population. Secondly, 3.6% of patients had either primary angle closure or were primary angle closure suspects. This means that the population had either synechiae or occludable angles on gonioscopy. So if we then look at the prevalence of the primary angle closure disease spectrum, that is the entire spectrum including PACG, PAC, and PACS, the prevalence in the RP population is 5.9%. And this is clinically important because we do perform dilated fundus examinations in these patients frequently, and it means that almost 6% of these patients would benefit from a YAG peripheral iridotomy. Now, how does this data compare with the population data? So if we look at Indian population-based studies, the prevalence of primary angle closure glaucoma in people over 40 years is 0.8%. We then looked at our RP population, identified those over 40 years, and calculated the PACG prevalence to be 3.8%, which is much higher than that in the general population. Hence, there does seem to be an association between RP and primary angle closure glaucoma. Moving on to the second part of our study, now the identification of glaucomatous damage in RP is challenging, and this is because both diseases cause RNFL thinning on OCT and visual field loss on perimetry. Hence, we need a different biomarker for glaucomatous damage in RP. We know that RP is a disease of photoreceptor degeneration, and in contrast, glaucoma is characterized by retinal ganglion cell apoptosis. Therefore, our hypothesis was in eyes with RP and glaucoma, the inner macula on OCT imaging will be thinner when compared to eyes with RP alone. To explore this hypothesis, we conducted a study to compare the thickness of different retinal layers on macular OCT in eyes with RP and glaucoma with those of only RP. So this was a case control study. Our participants were the cases who had RP and glaucoma and were compared to controls who had only RP, and the two groups were matched for vision and central foveal thickness. We excluded all patients with any maculopathy or poor quality OCT images. All patients underwent a SDOCT macula on the Heidelberg system. In addition to the total retinal thickness and the individual retinal layers, we also calculated the ganglion cell complex, which is nothing but the ganglion cell layer, the RNFL, which includes the axons of the retinal ganglion cells, and the inner plexiform layer, which includes the dendrites of the retinal ganglion cells. Statistical analysis was performed by mixed effect models, and the two groups were compared after adjustment for the use of 2 eyes per patient. Coming to our results, we had 21 eyes with RP and glaucoma, which were compared to 26 control eyes. They were demographically similar. The cases with RP and glaucoma had a higher intraocular pressure and a higher cup-disc ratio compared to the controls. Moving on to the macular OCT measurements. So the values in black shown here are those of the controls, and the values in red belong to patients with RP and glaucoma. When we looked at the total retinal thickness, there was no difference between the two groups in any of the ETDRS regions. However, when we looked at the ganglion cell thickness, in the inferior nasal perifoveal sectors, the cases, that is the patients with RP and glaucoma, had a significantly thinner GCC compared to the controls. When we looked at the outer retinal thickness, again, there was no difference between the two groups. Therefore, to conclude, almost 6% of RP patients have primary angle closure disease, and hence a gonioscopy is warranted in all patients with RP. Secondly, eyes with RP and glaucoma have a thinner GCC layer in some of the perifoveal sectors on macular oct, and this could be a potential biomarker for glaucomatous damage in RP. And lastly, future work should involve longitudinal studies of patients with RP and glaucoma to monitor the progressive changes on macular OCT. Thank you.
Thank you, Dr. Zia. We are ready to take any questions.
Zia, sorry, may I ask a question?
Go ahead, sir. Go ahead.
Zia, these layers that you delineated, was it automatically done by the proprietary software of Heidelberg which you used, or was it done by some other software which you included?
So the Heidelberg software gave us each of these individual layers, which is automated. When we calculated for each individual layer, because the layer is so much thinner and the layers are related, the individual layers had a significance, but not as strong as when we looked at the entire complex. And the disease is not really different in the different layers. When you have ganglion cell loss, you will have secondary loss of the axons as well. So the statistical significance...
I'm leading to something else. This is something, the lines are drawn by the computer, which
Yes.
is subject to error. Yes. So
Yes.
is there any way in which you could override and correct over that?
Yeah, so for every image, we did look at the segmentation that the automated software had performed. If there were any problems with the image quality, we did see that the segmentation was affected and we deleted those images. And then we actually arithmetically calculated the combination of the top 3 layers to determine the ganglion cell complex. That is how we did it for our particular study.
Okay. And again, the thickness and estimation or guesstimation was done by the computer algorithm, right?
That is
There was
correct.
no way you could intervene in that, right? If you
No.
found it genius.
So we did not change any of their sort of estimations. But if we found that there was a problem based on looking at the B scan image, we removed those patients if there was a problem with the segmentation.
Because your central foveal thickness
Yes.
itself was relatively high compared. It was in the normal range. What you showed.
Yes.
So this may not really represent.
Yes.
What is
So I will explain. So in our population, if you look at the vision, it was around 612 to 624. That means that these patients didn't actually have a lot of cone loss. And because the central fovea is predominantly cone related, these were patients who predominantly had photoreceptor loss in the rods, but it hadn't spread to the cones. This central foveal thickness also is deficient of ganglion cells. So you don't get glaucomatous damage as well in this area. And that is the reason we chose this zone as a sort of parameter to match the two groups.
And did you correlate this with visual also?
So we intentionally didn't do that because say we had two groups, which the two groups were matched for visual fields. It is possible that the group with only RP had actually more advanced RP, and the group with only cases had less advanced RP, and some of the visual fields was attributed to the glaucomatous damage. So we didn't use that particular parameter to compare it to to match for visual fields. But when we did look at the final analysis, the two groups had similar mean deviation and similar visual field index across the two groups, but they were not intentionally matched for in the study.
Very nice study. Thank you, Dr. Zia. Dr. Manoj, you have any questions? Please go ahead.
Yeah, and Dr. Zia, as even my comment on the first round itself for this same paper, if you remember right, I told you this is more sensitive only to the Heidelberg platform. Though the data analysis is good, but it's all automated computer generated analysis. We will like you to, because the reputation is very poor in this patient because of the fixation losses. So you need to work on that and get us some more data.
Yes. That is part of our longitudinal assessment. We intend to even look at manual measurements of, say, the ellipsoids, length of the ellipsoid zone, etc., to monitor these patients in future.
Okay, thank you.
Thank you so much.
It would be a good idea if you match these RP with glaucoma, with RP without glaucoma, and also with glaucoma without RP, so then you really have
a good complete spectrum.
Yes. Thank you, Dr. Zia. We wish you all the very best for your future. Thank you.
Thank you so much, Dr. Zia.
Introduce the next speaker.
Moving on, I'd like to introduce the next speaker. We have Dr. Purna Chandra B.
Good morning, I'm here.
Good morning, sir. You can start sharing your screen. We can see you and we can hear you.
Yes. Hope you can see my screen.
Yes, we can.
Just let me know once it's seen in the full screen mode.
It is seen in the full screen.
It is seen in the full screen mode. Am I allowed to start now?
Yes, sir. Please go ahead.
Okay, fine. I'll start. So, good morning, everyone. So, I'll be presenting my paper on a comprehensive approach to and exploring therapeutic potential in retinal dystrophies. No financial interest. As we know, field of genetics is evolving at a very high pace. Eye is the second only to the brain as an individual organ in its frequency of involvement with genetic disorders. And almost 50% of pediatric blindness is due to genetic etiology. Clinical picture can be sometimes overlapping. It may range from a mild clinical manifestation in case of stationary night blindness to a severe onset early retinal degeneration in case of Leber's congenital amaurosis. So these are some of the complaints with which patient present: night vision problem, normal vision, but tunnel vision. Some have central scotoma, some are allergic to light, they are photophobic. And some parents bring their kids stating that they are not doing well in the school. And some are worried, I have this condition, it's okay, but will my children get affected? So these are the problems what they face. What we are doing as of now is your counseling, genetic counseling we are offering. We are offering low vision services, photochromatic glasses for photophobia, and we are monitoring the disease. So due to its rarity, the clinical models of dystrophy has not been studied extensively in India and needs to be comprehensively analyzed along with the disease phenotypes. Ours was an observational study approved by Institutional Review Board. Informed consent was taken from patient and by guardian in pediatric patients. A total of 45 patients in the age group of 5 to 31 years were studied. There were 31 males and 14 females. All patients underwent comprehensive evaluation, including imaging. For pediatric patient, we used handheld OCT. Electrophysiological tests were carried out wherever required with strict adherence to International Society of Clinical Electrophysiology of Vision. So coming to the genetic testing part, we use next generation targeted sequencing using a panel of 212 retina specific genes, and we look for variant of interest. So we found 45 different pathogenic mutation in all these patients, and some of them were unreported, which are highlighted in the red. So among these, as I told, 9 mutations were unreported. These nucleotide changes were not present in 100 normal controls analyzed. 36 variants were previously associated with retinal dystrophies. So we had 9 novel mutations with existing spectrum of gene mutation, which are from the Indian cohort. So it provides further information on genotype-phenotype correlation for retinal dystrophies. So what does all this mean to patients? So let me explain the same by these two case examples. Both these patients presented with blurred vision. Fundus appeared normal clinically. The first patient had a very subtle change at the foveola, but however, OCT picked up subfoveal cavitation. When he did a fulfilled ERG, the dark adapted responses were completely normal, but if you look at the light adapted response, it was unrecordable. The second patient had obvious atrophic changes in the autofluorescence image. Fovea also showed thinning and outer retinal loss. However, fulfilled ERG was near normal, except for maybe a mild reduction in dark adapted responses. Why there is so much of functional difference, though both look similar clinically? The reason being the first case is a case of achromatopsia caused by GUCY2D mutation gene. And this is a stationary disorder. Patient is born like that and is going to live like that. And we need to counsel and we need to advise the career according to his needs. The second case is a Stargardt's disease. This patient had a mutation ABCA4. Though it may look functionally good now, it is likely to progress by taking some measures like avoiding bright light, avoiding vitamin A supplements. To extent, we can arrest the progression. So coming to the future, we know future is definitely going to be dominated by gene therapy for these trophies. Basically, gene therapy involves in injecting adenoviral vector, which carries a corrective gene, which gets inserted into the host cell where it's expected to function normally. So how to produce this AAV? It's a very complex process where plasmids will be generated first, which will be transfected into HKE cell lines. These will be harvested 65 hours later, and this will undergo multiple purification process, finally resulting in an AAV particle. This is the electron microscopy view of the real AAV particle. So we built a mouse retina model where we injected subretinally into this mouse model of the lag gene, which codes for beta galactosidase enzyme, and a separate ethical clearance was taken for this animal study also. This is the fundus photo of the mouse. We can see the injection site, and we also used fluorescent bead tracking system just to see whether they have properly migrating in the subretinal space. So 2 months post-injection, these eyes were harvested. And if you look at the negative control eye and the injected eyes, there was a very good staining of the beta galactosidase, which is quite promising. That means the uptake is very good, whatever the vectors we injected. So I would like to conclude by under these headings. What we knew so far was diagnosis of dystrophy means almost end of the road to patients. What this study adds is more database, and we had 9 novel mutation. It's indicating that somewhere the Indian database for mutation may be different from rest of the world. We need our own database. And subtle changes, as I showed in those 2 case examples, can make a big difference in the patient management and how we counsel. Rodent model was also studied in our study. The future will obviously be to carry out the first in India human gene therapy. Thank you so much.
Thank you, Doctor. If there are any questions, we'd like to take them now.
Dr. Purnachandra, you mentioned in your last slide that you would like to, create a greater database of the Indian genome and the Indian defective genes so far as all these retinal dystrophies are concerned. So towards that, what is your institution doing?
So as of now, like all the patients which have come to the dystrophy clinic, first they will be obviously undergo thorough clinical evaluation just to confirm that it is dystrophy, not something else which is treatable conditions. And these patients undergo a thorough genetic counseling first, just to make them understand what exactly genetic testing is going to offer. After that, there is a cost limiting factor when it comes to genetic testing. So as of now, as a study part, we are doing all this for free from our institute for all these patients, and we have generated data for almost more than 1,500 dystrophy patients. Eventually, since I mentioned the next generation targeted sequencing, once we have this large data, we can do a specific mutation screening, which will bring down the cost like anything. For that, we need to have this data, what we have generated.
No, but are you also parallelly collecting and screening normal people and building up the normal database?
Yes, yes. There is an exact genome database which is called, in which we have collected all the normal database. Once we generate any report, we have to look for the significance. For example, if we detect one mutation, we need to look at the frequency of that mutation happening in a normal population. So then only we'll compare with our mutation becomes significant.
But that
Otherwise,
means
if I undergo genetic testing, I may have some mutation. The issue is how significant it is. We have taken care of that part.
But that means you must have collected, against 1,500 people with retinal dystrophies, you probably must have collected what,
Yes,
15 lakhs of normals? Or
no,
how?
not 50 lakhs as of now, sir. Of course, in India we do have some logistics. It's around a lakh. It's around a lakh. Okay. But and also one more thing, sir. This is our thing what we added. But there's in the past also exact genome database they've already collected. It's not from our institute. In the genome, whole exome genome, there is a database for Indian patients also.
Yes.
That's our data.
So you probably need to tie up with those centers where they already have
Yes.
the
we,
genome.
we, we do have tie-up with Moorfields Hospital, and all of them have approached, because once we presented our Indian data, they were very much happy to see this variety of mutation, novel things and all. Even they were very happy to collaborate, sir.
Manoj, please ask your questions.
Yeah, Puran Chandra, good work as usual. Just two comments or two maybe suggestions. Can we extrapolate this data to most of the retinal dystrophies? That is one. And again, comparison of this data from Indian subset as well as overseas data should be, is the need of the hour. Then only we'll have more light on this particular research as how we are going to take this forward.
Yes, I'll answer that. So the first part you told, definitely this data can be extrapolated, because if you know that whenever we report the genetic, they'll be reported as pathogenic or variants of unknown significance.
Absolutely agree.
Yeah, once it is reported pathogenic, automatically it becomes relevant to other dystrophies also. And the second part, when you told, which is a very good point which you made, that if you look at if you comparable with the Western data. So here we need to compare it, because for Indian database for mutation, I told it may be different. For example, if someone designs a gene therapy model for something in the US or Moorfields, we can't come directly and inject that patient. Here the mutation may be slightly different. For
that
example,
is where the challenge is going to be.
Exactly. It's a good point you made. Thank you.
Purna Chandra, before we conclude, just one comment that please do a good spell check of your presentation. The spelling of achromatopsia was wrong. So
Okay,
similarly,
sir.
just
Yeah.
check out all your spell checks. Okay, go through all the slides again.
Okay, sir. Okay.
We won't hold it against you for that spelling mistake. Don't.
I didn't. Okay, thank you, sir. Thanks for the suggestion.
Thank you.
Thank you so much, panel. And I'd like to thank the speakers as well as the judges for being a part of this